So this is a recall of the organelles chunk map that I've created in the aim step or pre-study. Unfortunately, the recording for the pre-study was it's still on Loom and it cannot be downloaded, so I guess I'll just be drawing the chunk map and then comparing it with the actual map. And so what I remember is I'm trying to recreate the backbone first, so it's synthesis of biomolecules that leads to replication and also protection. Protection. Okay. And in this process, after synthesizing, you will have transport. I can't spell transport. Jesus. And also ATP synthesis by our good friend, the mitochondria, the powerhouse of the cell, lovely. And transport includes the Golgi apparatus and nuclear pores, because it's like go through the nuclear envelope. And then this is I'm not gonna write it down. Modify, process, modify, sort, package, and then transport the products of the ER, which we will get to in a second. And knowing what biomolecules are, I know nucleic acids are probably biomolecules too, but I couldn't be bothered to put it here since it belongs here. I think it suits here more, so I left it up. But the three main ones that everyone would think of immediately is probably carbohydrates. Then you have lipids. Proteins here. Then lipids. There's a reason why I have to put it here, which we'll get into a second. And I'm just gonna so for thylakoid it's light dependent, and for stroma it's light independent, so I drew a moon and a sun. Definitely not Pokemon, but whatever. And then for proteins it's RER, which I would say this direction, because RER contains ribosomes which synthesize the proteins, and then RER, the cisterna lumen of the RER, facilitates the folding of the polypeptide chain to form the protein. So I think they are both like different categories, so I didn't just put it like ribosomes here. I guess that makes more sense for me at least. And then protection, it will be like protect. Removal of harmful substances, which is SER, which one function of it is to synthesize lipids, phospholipids and steroids, but it also is involved in the detoxification of drugs and poisons, and so it falls under here. And the reason why I created this even in the first place is actually for lysosomes, which undergo digestion, autophagy and autolysis, and these two are involved. I don't feel like drawing it, but screw it. So it involves the fusion of membranes to release the acidic hydrolytic enzymes, and the reason why I drew it like a lemon because it's acidic pH. But anyways, and then this one is release cell contents, like all together. I can't pronounce the word, but I'll write it out. So I drew a jail cell and releasing content, which is a YouTube logo. Interesting, I guess. So it's release cell contents. Okay. So the other one, protection, is like having a selective barrier, which is like cell wall or membrane, and also the nuclear envelope, which I want to write down as a double membrane because I only remember it's chloroplast and mitochondria. Also from the symbiotic thing, endosymbiotic, some shit, whatever. Boom. And application involve two things. One is, okay, this is the one I can't remember. I remember the individual parts though, but I don't remember the chunks. We got chromatid. Oh wait, shit. And this is used for DNA replication, synthesis, or I rechunked it already. I gave up. So this one will be nucleolus, and this one will be nucleoplasm or some shit, and yeah, nucleus, protect, hereditary material from metabolically active cytoplasm. Boom. Okay. Okay, this part is a bit iffy, but otherwise, yeah, that's about it. I'm just gonna go check and open up this, and as you can see, I think I did pretty. Nope, I missed out some shit. Shit. Centrioles. Oh shit. Centrioles. It's actually centrioles mostly. What? Centrioles. There's nine triplets. So I drew the cloud9 symbol, perpendicular to each other or 90 degrees, arranged in a ring, act as microtubule organizing centers that help in the they appear in prophase to bind to the kinetochore proteins for mitosis and meiosis. Whoa, okay. And with that, yep, I guess this is the end of the retrieval session. There are other ways to retrieve, such as using your practice questions, which I will do for the next retrieval session, and also teaching, which I don't think I'm teaching now. I'm just retrieving the mind map as a brain dump, or also generating questions which I don't do, or variable addition, which is adding like variables to the question to change the question itself. And with that, adios.